Now for part 2 of Health, Genes, and Family History, where we focus more closely on how genes affect health. In this video lecture, we'll look at chromosomal disorders, in which missing or extra chromosomes cause disturbances in physical and mental development. We'll briefly look at two of those conditions, Down syndrome and Turner syndrome. We'll talk about gene disorders in which inherited mutations directly cause health problems. We'll look at the difference between autosomal dominant and autosomal recessive mutations, as well as sex-linked gene disorders. In complex polygenic gene defects, in which inherited gene or gene combinations interact with the environment that lead to development of certain diseases, and inheriting cancer risk, in which damaged mutated genes are found that cause or allow cancer to form, and these mutated genes are referred to as oncogenes. Chromosomal disorders include Down syndrome, in which an extra copy of chromosome number 21 is inherited, and it appears that this condition increases with increasing maternal age. If this is a chromosome map, which we have all 23 pairs of chromosomes, 22 pairs of autosomes, and the X, and in this case XX, so what we're looking at is a girl. The number 21 chromosome is shown to have 3 copies, and that leads to Down syndrome. Down syndrome has a familiar presentation with a broad forehead, a flat brow. There are mental and physical defects. Individuals born with Down syndrome are able to lead healthy lives right now into their 40s, given the proper resources. Turner syndrome is a chromosomal disorder that affects females only. And in this case, there is either a complete or partial absence of one of the X chromosomes. So we're looking at a condition that does not affect the autosomes, but rather it affects the sex chromosomes. In this case, we're either missing an X or it's, in this case, so small we can't see it. Characteristics of individuals with Turner syndrome include underdeveloped body, short stature, congenital heart defects. Gene disorders come in two major types. We have autosomal disorders, which occur on one of the 22 non-sex chromosomes, also known as autosomes, or they can be sex-linked gene disorders, in which there is a mutation in the X or Y chromosome. And if we're looking at the sex chromosomes, what is the sex of the person with this particular combination? This is Caster Semenya. I don't know how many of you have heard of her. She was a relatively unknown young lady who came to fame when she won the gold medal in the women's 800 meter in 2009 Olympics. But she apparently won by such a large margin and apparently with such effortlessness that her competitors felt that, well, she shouldn't have been allowed to compete. They thought she had a very masculine appearance, and some type of genetic testing was demanded. I don't know what the final results of those tests were, whether she indeed had a Y chromosome, whether she was a double XY, but I do know that she was allowed to compete in the 2012 Olympics. I'm not sure what her status is for the 2016 Olympics, but I imagine it's on the internet somewhere. In dominant gene disorders, a single copy is enough to cause the disease in all carriers. If we look at this diagram, we can see the possible combinations. If we have an affective or carrier father and a non-carrier mother. The mother has 2 small n's, meaning she has not inherited the dominant gene from either parent. And in the father, we have a large D, meaning he has inherited the dominant gene or affected gene from one parent and a non-affected gene from another parent. The father is affected. These are the 4 possible combinations: an affected female, a normal male, an affected male, and a normal female. So when it comes to dominant gene disorders, each child has a 50-50 chance of inheriting the disorder. Again, in a dominant gene disorder, you only need 1 copy of the gene to cause the disease. Examples include porphyria, which is a blood disorder. certain types of dwarfism, and the one I'll spend a few minutes speaking about is Huntington's disease, a degenerative disorder in which the nervous system is affected and results also in early dementia and death. Chris Furby is in a race against a disease that killed his mother, his aunt, and his grandfather. He also has inherited the disorder, and he is working on a film to raise awareness and to look for a cure. I also ask you to listen to this podcast. We'll talk about it in class, in which Charles Sabine, a foreign correspondent, I should say a former foreign correspondent for NPR, found out that he had the gene for Huntington's disease. Talk about the decisions he and his wife made to have a child. Recessive gene disorders come in two types. We have what's called the autosomal disorder and the Xlinked, or sometimes referred to as sex-linked disorder. In autosomal recessive disorders, 2 copies are required in order to produce the trait in both males and females. In this picture, we have both parents being carriers, and if this is the possible combinations for their children, they have a child 1 out of 4 chance of not carrying the gene, a 50-50 chance of being a carrier, and the carriers generally do not exhibit any of the traits of the condition, and 1 out of 4 chance of inheriting the gene from both mother and father and being affected. In X-linked disorders or sex-linked disorders, 1 copy is sufficient to produce the disease in males. 2 copies are required in females. Most autosomal recessive disorders are rare. Examples include Taysachs disease and cystic fibrosis. Taysachs disease is a disease that's found in Eastern European Jews. It's characterized by progressive degeneration of the nerves, leading to paralysis and death, usually by age 3. Cystic fibrosis is probably the most common genetic disease in the United States. There is no cure for it, but with medication, therapy, proper cystic fibrosis nutrition, individuals may live into their mid-30s. What happens in this disease is mucus builds up in the lungs, they have trouble breathing, and there's also malabsorption of nutrients because the pancreas doesn't produce the enzymes that are needed to help break down food. But again, with proper health care in 2009, the median survival age for individuals with cystic fibrosis was found to be in their 30s. Sickle cell anemia is another example of an autosomal recessive disease. Instead of having these nice round red blood cells, what happens with sickle cell anemia is that due to an inherited defect from both parents, these red blood cells form a sickle shape. If you inherit 1 copy of the gene, you're said to have the sickle cell trait. This is thought to protect against malaria. 2 copies mean that you have sickle cell anemia. These sickle-shaped cells clog the arteries and can lead to stroke and severe pain. In X-linked recessive disorders, males who inherit the affected X from the mother will have the condition. Females will have to inherit an X from both their father and their mother in order to have the condition. If they only inherit 1 affected X chromosome, they will be carriers because they have this other X to protect them. Examples include hemophilia and color blindness. I'm hoping that when you are looking at this picture, you are able to see a number. If you are not, please see me in class and we'll talk. In complex-polygenic gene defects, we're looking at a disease that results from multiple inherited genes interacting with environmental conditions that may lead to a disorder. Last but not least, we can talk about inheriting cancer risk. Now, the strength of the genetic link can vary. You can have a strong link between the gene and the disease. For example, in colon and prostate cancer, there appears to be a relatively strong genetic link. So, for example, if you look at your family tree and you see a large number of individuals inheriting colon and prostate cancer before they turn 50, that suggests a strong genetic link. With breast and ovarian cancer, there is sometimes a strong link depending on the type of cancer and the type of gene. Lung cancer is a type of cancer that appears to have a rather weak genetic link, but a strong environmental component. A normal gene that is vulnerable to damage and can lead to cancer is known as a proto-oncogene. It usually requires one or more mutations to convert a proto-oncogene into an actual oncogene. An oncogene is a damaged gene that is ready to cause cancer by either failing to regulate its own growth or ignoring regulatory messages of neighboring cells. We'll talk more about this at the end of the semester when we talk more about cancer. Some oncogenes may be inherited in an already damaged form, putting the individual at even higher risk for developing certain types of cancer. The one you may have heard a lot about recently is the BRCA or BRCA gene, BRCA1 and 2. What most people don't understand is that the BRCA1 and 2 gene code for tumor suppressor proteins. They code for proteins that help repair damaged DNA. They are related to suppressing tumor growth. But sometimes mutant varieties of these two genes produce proteins that don't protect as well. And they lead to an 80% increase in risk of breast cancer, and a 40% increased risk of ovarian cancer. These genes can be inherited. They appear to be more common in Eastern European Jews. Blood test that Angelina Jolie had is able to test for the risky varieties of these genes, for the mutated forms of these genes. I ask you to listen to this podcast about how breast removal can reduce cancer risk in some women. For class, we're going to finish the rest of this topic. I ask you to listen to the podcasts. We'll discuss genes and the psyche. We'll talk about genetic screening, a little bit about genetic therapy, and class discussion will include questions like, if you test positive for the Huntington's disease, should you be allowed to have children? If you or a loved one have the BRCA defect, would you have a prophylactic mastectomy? And probably whatever else I can come up with. Thank you.